Welcome back. We are now going to go to the urinary system. We're winding down. A few more chapters. We're getting close to the end of the term. We will now deal with the first part of the urinary system. And let's take a look at the function of the main organ for the urinary system, and that will be your kidneys. Your kidneys are a major excretory organ, and they maintain the body's internal environment by regulating the total water volume, the total solute concentration in water, regulating ions concentration in the extracellular fluid, ensuring long-term acid-base balance, and excreting metabolic waste. Okay, that's pretty much what most of its job is. And of course, one of the not really so unusual of functions of other organs, they there is also something hormonal in what they're doing. They produce a hormone which you call erythropoietin. It regulates blood pressure and renin, and therefore would regulate also red blood cell production. So it is also, to move on, responsible for activating vitamin D, carrying out gluconeogenesis if the body needs to. And the kidneys are pretty much part of the urinary system, which also includes the ureters. These are accessory organs, the urinary bladder, which is a temporary storage reservoir for urine, and then urethra, which is the one that transports urine out of the body. And this is pretty much how the gross anatomy would look like in a model. Take note that your kidneys are located outside of the body cavity, behind the peritoneum. Therefore, it is retroperitoneal. Okay, and this is actually your model from the cadaver. So let's take a look more at the gross anatomy of the kidney. It is retroperitoneal. It is in the superior lumbar region located between T12 and L5. The right kidney is a little bit lower, crowded, and so pretty much, uh, um, well, it doesn't really have much impact on what it does. It's just a little bit lower because it's crowded. And then for both kidneys, and it's not showing this picture here, there is a cap like gland that's capping over, sitting on top of each kidney, and that's what you call the suprarenal gland or your adrenal gland. And they do have a hilum, which is also um, sort of like the one that is um, um, on the inside, which is with well, actually the shape is concave, all right, and then it has a convex lateral surface, and then the middle part is actually what you call the hilum, and that's actually the concave part. The ureters are connected or is actually like exiting out of the renal hilum, together with the blood vessels, the lymphatics, nerves, pretty much, and blood vessels, if I have not mentioned it. And this is a position of the kidney, right, from a different view. So there are three layers of supporting tissue that surrounds the kidney. You have the renal fascia. It anchors the outer layer and it's made of dense, fibrous connective tissue. Then you have your perirenal capsule. It surrounds the entire kidney. It is a fatty cushion, and pretty much your kidneys does not have any mesentery, or because it's outside of the peritoneum, it doesn't have any mesentery or any um, membrane that would anchor it to the body wall. It is only the fat. Okay, so whenever um, we lose a lot of weight, and really, if we're going to be getting to starvation diet and lose a lot of weight in that area, our kidney could actually like drop. Okay, and that would make your well, the ureter kink, and there's going to be like an accumulation of urine that's not going to be emptied out, and so that would lead to failure of the glomerulus in the kidney. All right. So pretty much the third layer is actually the fibrous capsule. It prevents the spread of infection to the kidney. Instead of a visceral peritoneum, it would have a capsule. This is actually picture showing you how the kidney is retroperitoneal. The peritoneum is this brown line right here, okay? Right? No, not that one. The one on top, the white line. The peritoneum is that white line. This is pretty much the area that would cover on top of the kidney. And this entire red area here is a cavity, okay? We, you're not, it is not showing the rest of the other organs in the peritoneal cavity. So, your kidneys pretty much are on protected by the thoracic cage. It's fat, the perirenal fat, provides proper cushioning. The lower parts of the kidneys are susceptible to blunt trauma. So you could see that the lower part of the kidney right here is not much protected by the thoracic cage. So, for instance, any falls, major vehicular accident, or contact sport injuries that would pretty much like uh, be in contact with the kidney can actually injure it. So the renal artery is very vulnerable to injury from rapid deceleration during uh, car crashes, and it could lead to lacerations or thrombosis, blood clot. Hematuria, which is blood in the urine, is an important sign of such trauma. And surgical treatment, okay, after a thorough scan, might be required in order to find out exactly if there was a tear in the blood vessels of the liver. I mean, of the kidneys. Okay, so let's take a look at the internal anatomy. The kidney, just like any other solid organ, would have two sections. The first section, the middle part, this part here, that's really very busy, okay, is what you call the medulla. And the outer section, okay, which is really quite smooth and not so busy in this picture, is actually your cortex. It is, look, the cortex is all superficial, and the medulla is actually deeper into the cortex, and it carries the renal pyramids, the papilla, the renal columns. These are the renal columns right there. The pretty much the um, the pyramids is actually this section here, the dark brown area that looks like an inverted pyramid. And you have your renal pelvis, which is a funnel-shaped tube that is continuous with the ureter. So this is the renal pelvis right there, or right here, this is the renal pelvis. The renal pelvis receives urine from the calyces. Okay, you have the major calyces. These are the big ducts right here that opens and receives urine from the minor calyces, and these are your minor. The minor calyces are the ones that are directly emptying urine from the papilla, okay, into the into the pelvis. So, some homeostatic imbalances. Pyelitis is the infection of renal pelvis and calyces. And pyelonephritis is infection or inflammation of the entire kidney. The infections in females are usually caused by fecal bacteria that's entering the urinary tract because we have a very short urethra, and bacteria could enter into that due to poor hygiene. And then you have several cases that can cause swelling of the kidney and abscess formation, and pus may fill pretty much the renal pelvis. And pretty much this is actually like one of the extreme cases in pyelonephritis already. And if it's left untreated, the kidneys may be totally damaged. Usually it is successful. Well, The treatment would be like successfully with antibiotics. So let's take a look at the blood supply. Well, kidneys would be cleansing blood, and that's the purpose of the kidney, to receive the blood and to filter it and then empty it out already filtered of whatever it needs to be filtered out. So the renal arteries, so these are the red ones, the renal arteries deliver about uh, 1,200 ml of cardiac output to the kidneys for every minute. You could just imagine that, 1,200 ml, 1.2 liters per minute. That's exactly how hardworking our kidneys are. And you think your heart is really like hardworking because it has a very effective cardiac output. Well, the renal output is also very effective. The renal outflow starts from the renal artery, and the renal artery is right here. Then it goes into the segmental, the interlobar, which is actually the one that is in between the lobes, right there. Those are the ones that are running in between also the pyramids, right into the renal columns. Then you have into the interlobar, and then the arcuates, okay. Then from the arcuates, blood flows into the interlobular, or the radiating blood vessels, arteries, and then into the afferent arterioles and into the glomerulus. So pretty much it goes back from the glomerulus. Okay, once it has been filtered from the glomerulus, it goes back, and in the peritubular arteries, it goes back into the cortical radiates, into the arcuates. Okay, that's actually also the same thing. They have the same name. Both blood vessels, veins, and arteries would have the same names, and they're called arcuates because they are forming an arc, like an arc right there. From the interlobar, they will split into two arcuates right there. So from the arcuates down into the interlobars, and then directly into the into the renal veins. There's no segmental veins. There is segmental artery, but no segmental veins. And the nerve supply is actually a sympathetic fiber from the renal plexus. And pretty much this is a very good cheat sheet. You can actually see from the aorta to the renal artery, this is exactly how the blood flows. And then it gets into this part here that would have broken lines, is already the part where it's getting into the nephron. These blood vessels right here are already the blood vessels associated with the nephron's blood supply. And then from the nephron, it moves out again and joins again the mass of the kidney, and they are already outside of the nephron, from the cortical radiate vein all the way down and then back into the inferior vena cava. So you see here, the renal arteries carry blood that's unfiltered, and the renal veins carry blood that has been filtered already. So, who filters it? Well, there's a unit for filtration on the kidney. The kidney is just the organ. There are smaller units, organ system made up of different types of cells in an organ, which you call the kidney, which you call the nephrons. The nephrons are structural and functional units, and they are the ones that form the urine. There's about 1 million of them per kidney. And they do have two parts, the renal capsule and the renal tubule. This is your capsule right here. It looks like a cup, okay? And then connected to the renal capsule, you have convolutions of tubules. So are your renal tubules. So there's only two parts to a nephron, the capsule and the tubules. The capsule is actually where filtration of the blood is happening, and then the filtrate travels right into the tubules. Okay, we're going to go into those in a few minutes more. All right, so Let's take a look at the renal corpuscle. Okay, the renal corpuscle right here. This is actually your entire renal corpuscle, and is made up of the glomerulus, which is this section right here. You have the blood vessel, a tuft of capillary that enters the cup itself, and it would have fenestrated endothelium. They are very or highly porous capillaries. They allow for efficient filtrate formation, and pretty much your filtrate is actually pretty much a plasma derived fluid that the renal tubules process to form the urine. And this renal filtrate is actually going to be filtered right through. So when blood goes from here and it enters there, then that's already what you call the renal filtrate. And take note that the filtrate is only plasma, okay, and some other minor components. But most of the formed elements and big proteins stay in the blood. And take note of the difference, okay, in the entry of the blood from a wide afferent arteriole and the exit of blood from into a narrower efferent arteriole. So the afferent arteriole, the one that receives, the one that draws the blood into the glomerulus, has a wider lumen, and then the efferent arteriole has a narrower lumen. And so if you're going to think of what the consequences is of this particular two measurement of these two tubes, and they are unequal, then you would think of what's going to happen right within the glomerulus if this is so. The entry point is wide, and therefore the gush of blood is really a lot. Meanwhile, the exit point is really narrower, and therefore the traffic is actually heavier here, and there's going to be a backlag here. So it prevents the what do you call this? The fast movement of blood as it enters. So it slows down because of this narrow exit point. So there's an accumulation of pressure right in this particular area here. And that pressure creates a hydrostatic pressure that would move the plasma and becomes part of the filtrate right into the tubules. Okay, so the glomerular capsule is also known to be the Bowman's capsule. This entire thing here is the Bowman's capsule. It's a cup-shaped hollow structure that surrounds the glomerulus, and it's made up of two layers. The parietal layer is this outer section right here. It's made up of simple squamous epithelium. The visceral layer is actually the one that would be abutting, okay, it abuts or is exactly touching the glomerulus themselves. It clings, the cells of the visceral layer clings to the glomerular capillaries, and they do pretty much like extend branching epithelial, what it calls epithelial podocytes. So pretty much these podocytes are cells that would have false feet. That's why they call them podocyte. And pretty much the extensions of these cells here terminate in food processes that cling to the basement membrane, and because they are intertwining with one another, they form what you call a secondary filtration mechanism, which you call the filtration slits. Okay, these filtration slits are between the food processes, and they allow the filtrate to pass into the capsular space. And so let's take a look at, the structure, more of the structure of the nephron. Let's find out pretty much what cells are found in each of the areas. And I mentioned already to you the Bowman's capsule. What's made up? It's made up of, well, the visceral part is made up of podocytes. The parietal part is made up of simple squamous epithelium. Let's take a look at the rest of them. Okay, this is actually your parietal layer, and that's actually your podocytes. Let's take a look at the rest of them. Okay, here you go. This is the parietal layer. They're made up of flat cells, simple squamous epithelium, and the visceral layer of the capsule. It's made up of podocytes. You could see the false feet of the podocyte. And once you get into the renal tubule, that's actually now starting from this part here. Okay, you now have different cells forming the renal tubule, which is about renal tubule for every nephron is about 3 centimeters long, only about 1.2 inches long, and it's made up of a single layer of epithelial cells. But many regions have unique specialized cells. But for the most part, we just when you look at the kidney cross section, you look at them and you see nothing really but just simple cuboidal epithelium, which happens to be like one layer of cuboidal epithelium surrounding those little tiny tubules. And they are made up of 3 parts. The proximal convoluted tubule is the one that is directly receiving filtrate coming from the renal capsule, and this is your proximal convoluted tubule. Okay, the proximal convoluted tubule is located in the cortex of the kidney. Then it goes down and joins in a sort of like a loop-like a tubule, and you call it the loop of Henle, and this goes down into the medulla. Okay, now right here, this big section right here is part of this part here. This section right here is the cortex, and this section right here, bottom, okay, where you have the arcuate. Okay, these 2 blood vessels right here are the arcuates, the arcuate artery and the arcuate vein, where I use those arcuates as some sort of like um, um, well, some sort of like a um, um, uh, well, it's to me something that is showing me where the what do you call this, the cortex and the medulla ends. Okay, where you see that those arcuates, that's supposed to be for me part of a map, a perimeter line that separates the cortex. Okay, above it, above the arcuates will be the cortex, and below the arcuates will be the medulla. So you could see that your loop of Henle is already in the medulla. And then it goes up and then joins again, continues on into a um, distal convoluted tubule. And the distal convoluted tubule continues on with what it's doing with the filtrate, and then it empties into the collecting duct, and then finally into the papilla. So your proximal convoluted tubule would have cuboidal cells with dense microvilli, and pretty much it has a good amount of surface area. They do have a lot of mitochondria, okay, functions in reabsorption and secretion, and they are confined to the cortex. And then you have these are actually the cells of the proximal convoluted tubules. Okay, they do have a lot of microvilli, a lot of mitochondria because of its function. Then you have your renal, your Henle, loop of Henle, the nephron loop. Pretty much this is the nephron loop, which is confined pretty much in medulla, and the descending limb of the loop of Henle right there. This is the descending limb, and then you have an ascending limb, so it's actually U-shaped. Okay, and between the descending and ascending, you have what you call a thin limb. Okay, for both of them. Okay, made up of simple squamous epithelium. Then you have, okay, this is a thin segment. These are showing you the simple squamous epithelium for the thin segment of the nephron loop. And then the distal convoluted tubule. The distal convoluted tubule is made up of cuboidal cells with very few microvilli. They function more in secretion than reabsorption. And therefore, they don't really need much of the many microvilli as much as the PCT, or the proximal convoluted tubule. This is confined to the cortex. Okay, this now is a cross section of a small area of the kidney. It shows you this part here, which is the glomerulus, and that area right there outside the glomerulus is actually your simple squamous parietal layer of the Bowman's capsule. And you have the glomerular capsular space, this empty space right here. It's filled with filtrate, and you have right here supposedly the proximal. Well, this is actually a proximal convoluted tubule, and this section right here is already part of the distal convoluted tubule. Okay. Okay, so now we have the collecting ducts, which has two cell types. The first type is what's called the principal cells. It has short microvilli. It's sparse. You don't have much of it. They do maintain water and sodium balance. And you have your intercalated cells. This middle part here is the intercalated cells. They do have abundant microvilli, and pretty much there's two types of intercalated cells. You call them A and B. Both help maintain acid-base balance of the blood. The collecting ducts. The collecting ducts receive filtrate from many nephrons. So this is very much like a big tube that receives processed urine from many nephrons. They run through the medullary pyramids. They give pyramids their striped appearance, and the ducts fuse together to deliver urine through the papillae into the minor calyces. Okay. So, are all nephrons alike? Well, they're supposed to be do. Yeah, there are, right? But there are different sizes. Therefore, because of the sizes of the nephron, and some parts of the nephron would be longer than the others, um, it gives them some sort of a differentiation or differences in their function. For instance, this little guy here, okay, where most of its body is a nephron, most of its body is actually in the cortex, is what's called cortical nephron. And 85% of our nephrons are almost entirely located in the cortex. Okay, and most of our nephrons are actually cortical. Now there's a handful, which is about 15%, okay, which you call the juxtamedullary nephron. This long, they do have long loop of Henle that extends all the way down throughout the entire length of the medulla. They really invade deeply the medulla. The ascending limbs have thick and thin segments, and they're very important in the production of concentrated urine. This is because the purpose of your loop of Henle is to reabsorb water. Imagine if you will have a long line of ducts that would be reabsorbing water, then the urine that you're going to be producing for that group of nephrons will be concentrated because you are actually reabsorbing a lot of water. This one here doesn't do that. All right. And what is important with the juxtamedullary nephron is that it does have a series of um, um, capillaries, okay, that looks like a ladder, okay, and they pretty much are the only one. We call them the vasa recta. These vasa recta pretty much are only present in your juxtamedullary nephrons. The cortical nephrons would have an extension of the papillary, um, the papillary blood vessels, capillaries, reabsorbing filtrate from them. So urinal tubules are associated with two capillary beds. So I've been mentioning something about vasa recta and peritubular capillaries. What are they? Well, see, as our urine is filtered right there in the glomerulus, and then it goes into right into the tubules. at some point in time, the body would feel that there's so much really that has been filtered through it, and they need to reabsorb back some of the things that is actually filtered, or else we're going to be dehydrated. You don't want the all of the filtrate to become part of the urine. So some part of the filtrate would need to be reabsorbed back, and that reabsorption is actually happening through the capillaries. Capillaries around or surrounding the tubules are the ones that reabsorbs the filtrate. And pretty much you have your peritubular capillaries right here, okay, in the cortex part, and some of them out here for um, reabsorbing blood coming from, let's say, the short segment of the loop of Henle for cortical nephrons, but you have an extra long vasa recta for the juxtamedullary nephrons. Oh, okay, so pretty much you could see that right here. Oh, I forgot to mention to you that inside one again of the capillaries in the nephrons is actually your uh, glomerulus. This is actually a glomerulus, okay, which is actually part of the nephron thing. So let's take a look and mm-mm. I must have accidentally turned on something that's going to uh well, right there, could see it. Something is actually being written out there. Anyway, so the glomerulus are capillaries that are specialized for filtration. They are different from any capillary beds because they are fed and drained by the arteriole. And pretty much it enters, or blood enters the glomerulus, this is the glomerulus, through an afferent arteriole and exits the glomerulus through an efferent arteriole. Okay, the efferent arteriole is the one that feeds the blood, or that feeds the blood back into the peritubular capillaries or the vasa recta. So, blood pressure in the glomerulus, inside the glomerulus, and I mentioned this already with you, um, becomes higher than the blood pressure right here in the afferent arteriole because of the small diameter exit point, which is the efferent arteriole, and it creates higher resistance right in this area here, and therefore the blood pressure right here is getting to be higher. So your peritubular capillaries are low pressure capillaries. They are very porous. They're adapted for absorption of water and solids. They arise from the efferent arterioles. They cling to adjacent renal tubules in the cortex, and pretty much they empty into the venules. So the vasa recta is actually very long and thin-walled. They're parallel to the juxtamedullary nephron, and they extend all the way into the medulla to reabsorb filtrate that is coming from the loop of Henle of juxtamedullary nephrons instead of the peritubular capillaries. And the function of this is to pretty much reabsorb uh, more of the um, filtrate in order to form concentrated urine. Okay, let's take a look at the juxtaglomerular complex. You know, when I was in college and I was learning the anatomy and the physiology of the kidney, I called that juxtaglomerular complex, because it looks like that's the way it is, the brain of the kidney. And pretty much, if you're going to take a look at how the kidney works, the work of the kidney pretty much, or how it is engaging in how it's filtering and when it's going to stop filtering. How does it know if we are too dehydrated already? How does the kidney know when our blood pressure is too low or too high? When does it slow down, or when does it need to really produce more urine? Does it do that? Does it know that? Yes, absolutely, it knows that. And the way they determine it is because of the specialized cells that are found in the juxtaglomerular complex. The juxtaglomerular complex pretty much is made up of three groups of cells. The first one is what you call the macula densa. This purple group of purple colored cells right here. And they are found in the ascending limb of the nephron loop. Then you have the extraglomerular mesangial cells, and this is actually like a triangular mass of cells right here. And then finally you have the granular cells, and the granular cells are this one here, and there's another one here. They are found in the afferent, on the walls of the afferent arteriole. They're kind of like smooth muscles, okay, cells. And this one here are tubular cells. So they do communicate. The macula densa communicates with the granular cells. That's exactly what they need to do in order to form urine, depending on the situation of the body. And how do they interact? Well, it is actually noted that the mesangial cells, the ones that are right here in the middle, are the ones that help channel through the information between the two of them. And we're going to be discussing that when we get back into the physiology, or when we get into the physiology of these two. So, macula densa, you could see it, okay, in your lab. They're very tall and they're closely packed cells of the ascending limb. They contain chemoreceptors. They will be sensing the amount of sodium chloride content in the urine, this part right here. So when your urine would be high in sodium chloride, then I guess, you probably either will have to get rid of that sodium chloride, or when the sodium chloride is retained right there, water will have to be taken in. So this one senses the amount of sodium chloride in the urine. And you have also the granular cells, or the juxtaglomerular cells, and these will be your smooth muscle cells of the arteriole. They act as mechanoreceptors to sense blood pressure in the afferent arteriole. They contain secretory granules that contain the enzyme renin. Okay, so renin plays a major role, and you remember in your renin-angiotensin system, renin plays a major role in the production of angiotensin, or conversion of and production of angiotensin, and pretty much is actually produced by the kidneys. And these are the cells that produce the renin, or secretes the enzyme renin. And then you juxtaglomerular mesangial cells. Pretty much they are interconnected with gap junctions, and they are thought to pass signals between the macula densa and the granular cells. Right, let's take a look now at the physiology of the kidney. Okay, 180 liters of fluid per day is processed. But actually 1.5 liters, less than 10% only, okay, 1% is actually formed and excreted. Imagine if all 180 liters of fluid processed daily, meaning filtered through daily, is actually part of your urine, then you're not going to be doing anything but sitting on the commode just to eliminate water. But that's not really happening, right? You don't want to lose all of the water that you have, or all of the plasma of the blood that you have right there. So we consume, okay, our kidneys consume about 20 to 25% of oxygen, okay, used by the body at rest. The filtrate produced by glomerular filtration is basically blood plasma without the proteins. And urine is produced from the filtrate. Urine is less than 1% only. See that? I mentioned to you this is 1.5%, that's 180 liters. This is 1.5 liters versus 180 liters, so about 1%, less than 1% of original filtrate. And they contain metabolic waste and unneeded substances. So let's take a look at the first process. There are three processes actually. You have what you call glomerular filtration, and it is happening right here in the glomerulus. And then you have tubular reabsorption, which is happening along the tubes, okay? And then the last one is secretion, okay? What are they? Well, tubular filtration is actually happening whenever blood coming from the arteries would be delivered into the glomerulus at such high pressure. Okay, remember that there's this one is actually wider in diameter, and therefore it has a narrower exit point, and therefore there's an accumulation of pressure here. And that high pressure pretty much would push the um, um, the hydrostatic pressure is the only one that pushes plasma out of the glomerulus and into the glomerular capsule. This is your glomerular capsule right there. And tubular reabsorption is pretty much where all of the filtrate going through here, can you imagine that? If all 180 liters in a day would be processed here by the glomerulus, you don't want to be losing all 180 liters of fluid. So part of it is being selectively reabsorbed. You can see that. And they are reabsorbed into the peritubular capillaries, okay, and the vasa recta also. So pretty much, some part of it also, once it goes back into the blood would still be selectively moving substances from the blood back into the filtrate to pretty much like get rid of some more stuff, okay? So glomerular filtration, right there on the top, right where you have your glomerulus, is a passive process, and there is no metabolic energy required. The hydrostatic pressure of the water, that just like what I told you, it, right through the filtration membrane, pushes it into the glomerular capsule, into the space. Okay, there's no reabsorption happening here. Reabsorption would start right there in the tubules, in the proximal convoluted tubules. So your filtration is happening in the Bowman's cap is actually an effective filtration. It has an effective, very effective filtration mechanism brought about by an effective filtration membrane. It is a porous membrane between blood and the interior of the glomerular capsule. It allows water and solute and smaller than the plasma protein to pass through. It normally no cells can actually pass right through. Okay, this is an enlarged section of the filtration membrane, which is actually a piece of that portion right there, and it has three layers. First layer is actually your fenestrated endothelium of the glomerular capsule itself, and this is actually your fenestrated endothelium right there. The basement membrane, which is fused basal laminae of two other layers, and then the food processes. These are your food processes. They form what you call filtration slits, where your plasma will actually be filtered through. So you are not expecting big proteins. Okay, there will be amino acids or maybe dipeptides that can walk through, but proteins and cells are not allowed to walk through. So ions will be walking through. All those that are small enough to walk through will do. And here's actually an electron microscopy of a podocyte. Imagine this, guys. A long time ago, we are just imagining it. Now we are actually looking at it. The actual podocytes makes us very makes us makes it a little bit easier for us to pretty much realize where it's going, where things are going. So the filtration membrane, and this is the filtration membrane right through here. Okay, well, you have big molecules or macromolecules, okay, that are stuck in the filtration membrane. They don't just get stuck right there. They are going to be eaten by the glomerular, engulfed by the glomerular mesangial cells. The filtration membrane allows molecules smaller than 3 nanometers to pass. Water, glucose, amino acid, nitrogenous waste, okay? Plasma proteins remain in the blood to maintain the colloid osmotic pressure. And it prevents the loss of all water into the capsular space. And you should have a little bit of a pull effect, and that's your plasma proteins pulling your water back, and so you can retain some in the blood. And the proteins in the filtrate the proteins in the filtrate indicate membrane proteins problem. So let's take a look at the pressure. What are the interacting forces, which is interacting pressures that are affecting the movement of filtrate in and out of the glomerulus? Well, the outward force pretty much will be right there. The ones that are going into the capsular space will be your outward force. So you have one big outward force, which is pretty much the hydrostatic pressure, okay? The force that promote the filtration formation. A hydrostatic pressure, which is GC, is essentially the glomerular filtration blood pressure, which is the blood pressure that is formed, the pressure that is formed out there by the gush of blood coming from the afferent arteriole. But then, remember, guys, it's going into what we call the capsular space. But capsular space is not empty. It has pre-existing renal filtrate that is already there. This is an ongoing thing. That's not an empty thing. Nothing is pumping here. So if actually you're moving fluid right through here, there are opposing forces that are already here, a pre-existing fluid, and the pressure of the fluid that is already in the right here is pretty much like what you call the capsular space pressure, okay, and capsular space pressure, which is I think it's probably on the next one. Okay, the inward pressure right there, those two, okay, will be your hydrostatic pressure. And this hydrostatic pressure, this one here, okay, is the pressure of the filtrate, okay, which is about 15 millimeters of mercury. So you have a pre-existing filtrate here that is pushing the water, preventing the 55 millimeter pressure, mercury pressure of fluid from gushing completely out. You have an opposing force, which is 15. So you deduct that. So 55 minus 15 is actually what you're going to have, about 40, right? Then you have also the colloid osmotic pressure. Okay, the colloid osmotic pressure is actually the pressure inside the blood, the proteins in the blood that pulls the water in and keeps the plasma in there. It is just like it has a suction effect, okay? So it prevents the blood from getting into the capsular space. So between these two, okay, if you're going to take a look at the net filtration pressure, it's right here, around here in this area here, okay? Your hydrostatic pressure, which is 55, and the capsular pressure, or the pressure in the capsule itself, which is about 30, okay, no, 15 rather, and the osmotic pressure, you add them too because they're actually the the uh, inward forces that is preventing uh, the outward force from fully forming the filtrate. You actually like deduct the inward force from whatever total hydrostatic pressure you have. You're going to have a total of only 10 millimeters of mercury pressure. This 10 millimeters of mercury pressure is the only driving force, the only pressure that would be driving the plasma into your into your what do you call this, into the tubules, and that becomes part of the filtrate. So that's not even really much, 10 millimeters of mercury pressure. That's why if a person doesn't take a lot of fluid, okay, it gets to be like throughout the entire day, gets into a mild dehydration or something, that person is not anymore going to go to the bathroom. If your hydrostatic if your hydrostatic pressure, which is supposed to be 55, drops down for any reason, and this 10 millimeter would be zero, and you're not anymore going to form urine. There won't be anymore any formation of urine. So your blood pressure, rather the movement, rather the filtration, okay, the net filtration pressure is dependent on many factors. Okay. Now let's take a look at the rate, or how fast this filtration is actually happening. See, glomerular filtration rate, or GFR, is the volume of filtrate formed per minute by both kidneys, and normally it's about 120 to 125 ml. That's a lot. It's almost like one graduated cylinder per minute. That's exactly how fantastic our kidneys are in cleaning our blood. And our GFR is directly proportional to the net filtration pressure, which is the primary pressure of the glomerular, of the primary pressure of the glomerulus, and then the total surface area that is available for filtration. The glomerular mesangial cells is actually controlled by contracting here. Okay, so whenever you have the mesangial cells, which is found right here in the afferent arteriole, if there's going to be a lot of pressure going here, okay, how do you think will you preventing the accumulation of pressure right here? Well, the glomerular filtration right in this section right here, it says here, will contract. And if it contracts, it's going to make the lumen of the afferent arteriole smaller. So when the lumen gets to be smaller, then the pressure, the oncoming pressure of blood going into the glomerulus becomes less also. That's how we control it through the glomerular mesangial cell. So how will the glomerular mesangial cell know that? That's got something to do with how it communicates with the macula densa of the juxtaglomerular complex. So, and the GFR is also directly proportional to the membrane permeability, okay, which your glomerulus is actually much more permeable than the other capillaries of the body. So constant glomerular filtration rate is important because it allows the kidney to make filtrate and maintain extracellular homeostasis. The goal of local intrinsic control, which is renal autoregulation, it will be to maintain GFR in the kidney. So GFR affects the systemic blood pressure. Increased GFR causes increased urine output, which lowers blood pressure, and vice versa. The goal of extrinsic control will be to maintain the systemic blood pressure. Okay, so pretty much your GFR affects systemic blood pressure, and the extrinsic controls of the GFR maintains the systemic blood pressure. And the intrinsic control of the GFR maintains GFR in the kidneys. So the nervous system and the endocrine mechanisms are the main extrinsic controls of the GFR. So let's take a look at the intrinsic control in which is renal autoregulation. Pretty much renal autoregulation maintains nearly constant glomerular filtration rate when the mean arterial pressure is within 80 to about 180 millimeters of mercury. Now, autoregulation pretty much is um, not going to happen anymore if it is out of range, which means autoregulation is only going to happen whenever your mean arterial pressure is within 80 to 180. Below that or above that, your renal autoregulation would stop. So there are two types of renal autoregulation. One is myogenic mechanism, and the other one is your tubuloglomerular feedback mechanism. Your myogenic mechanism pretty much is a process that involves local smooth muscle in the kidneys when it contracts. Okay, whenever it contracts and whenever it's stretched. Okay, now for instance, increased blood pressure, okay, would cause the muscles to stretch, meaning you have a lot of blood getting into your blood vessels. It stretches your blood vessels. It causes the muscles there to stretch, leading to constriction of the afferent arteriole. So whenever there is a stretch, the afferent arteriole will react to it because that's a mechanoreceptor. It constricts, and thereby restricting blood flow into the glomerulus, protecting the glomerulus from damaging high blood pressure. So if a person really has chronic blood pressure, or if the blood pressure, or if a person suffers from hypertension, and it causes the blood smooth muscles of the blood vessels to stretch out, it will automatically signal the mesangial cells to constrict, and therefore that point of constriction will minimize or restrict the blood flow into the glomerulus, thus preventing too much pressure accumulating in the glomerulus. Now, decreased blood pressure causes the dilation of the afferent arteriole, okay, which is a reverse, and would help pretty much like promote your information. Both would help maintain normal glomerular filtration despite normal fluctuations in the blood pressure. Now, the tubuloglomerular feedback mechanism, uh, this is actually a flow-dependent mechanism that's directed by the macula densa. You remember that your macula densa is sensitive to the filtrates concentration of salt concentration. For instance, if your filtration rate increases, the filtrate flow rate would increase. Okay, this leads to a decreased reabsorption time. When there is a decreased reabsorption time, the sodium chloride level will increase, because what happens during the reabsorption is pretty much when there's actually exchange of both water and salt, both of them will have to be reabsorbed. Water follows salt. But if the rate of urine formation is too fast, the sodium chloride reabsorption is not going to be as effective. It's not going to be reabsorbing as fast as it should. It causes then sodium chloride level to be higher in the filtrate. And a feedback mechanism would cause the constriction of the afferent arteriole, which lowers the net filtration pressure and the GFR, thereby allowing more time for sodium chloride reabsorption. So whenever there's actually an increase in sodium chloride in the filtrate, there's going to be a message about the. Remember that your macula densa is a chemoreceptor. It sends a message into the mesangial cells, and the mesangial cells will cause the constriction of the afferent arteriole. And so let's take a look at the extrinsic control of glomerular filtration, which is pretty much neural as well as hormonal. Now, take note that the purpose of your extrinsic control is to regulate the glomerular filtration rate to maintain systemic blood pressure. And the extrinsic control will override the renal intrinsic controls if your blood volume needs to be increased. Most especially in some cases where, in an accidental loss of blood, maybe because of hemorrhage or something. Okay, the extrinsic control is going to happen, and pretty much the control is brought about by the sympathetic nervous system. And under normal condition, pretty much, the renal blood vessels are dilated. Okay, and here, under normal conditions, your autoregulation mechanism, the intrinsic one, is actually the one that prevails. However, under abnormal condition, such as extremely low extracellular fluid volume, okay, the blood pressure, okay, is now dropping. There's going to be a release of norepinephrine, okay, by the sympathetic nervous system, and epinephrine released by the adrenal causes systemic vasoconstriction, which would eventually increase blood pressure, constriction of the afferent arteriole, which will decrease the GFR, blood volume and pressure increases eventually, okay, and these are all controlled by the sympathetic nervous system. And so this is your renin-angiotensin system, okay, which is the one that maintains the mechanism for increasing blood pressure. And we have seen this many, many times in when we were discussing our blood pressure, okay, in the blood vessel, okay, which shows us the three direct pathways to renin release by the granular cell. Okay, the first pathway is what you call direct uh, stimulation. I think there's actually one here. Not all of it has been taken. Okay, direct stimulation of granular cells by the sympathetic. There you go, granular cells. Okay, of the sympathetic nervous system. Okay, then direct stimulation of the macula densa. Okay, which is right here. This is actually where the macula densa is. So pretty much it would cause all of them. Pretty much would have an increase, would result in an increase in GFR and an increase in systemic blood pressure. This is your renin-angiotensin system. I would urge you really to review it again because we did this already. All right, so let's take a look at the regulation of the glomerular filtration. Other factors affecting GFR pretty much would be like a variety of chemicals. Adenosine, prostaglandin E, okay? Some cells make their own locally acting angiotensin 2. They reinforce the effects of hormonal angiotensin 2, okay? And right here you have the summary of regulation of GFR. You got the extrinsic control, and you got the intrinsic control or the renal or regulation. You have the purpose, the mechanism, and the operating conditions. Guys, this is pretty much a very good summary, very good way of reviewing. That's why I included this in the PowerPoint. So, homeostatic imbalances, anuria, abnormally low urinary output, which is less than 50 ml per day. This may indicate that glomerular blood pressure is too low to cause filtration. Okay, renal failure and anuria can also result from situations in which nephrons really stop functioning. Examples of this will be in cases of acute nephritis, transfusion reactions, and premature crush injuries. That's it, guys. I bid you well, and in another day, I am going to upload the second part of the urinary system.